So yes, my name is Talia and I research a cure for HIV at Cornell. And I'm really passionate about what I do. I've lived in Cambodia, India, and spent time in Botswana working in HIV and AIDS clinics, mostly with women and children living with HIV and AIDS, and those experiences have changed my life forever. And while I did study the Masters of Public Health, I endeavored to cure the virus so I could hopefully change millions of lives around the world. So, don't mind the theme is Star Trek, but the newest Star Trek just came out. Do we have any Star Trek fans in the audience? Okay, good. A few, thank goodness. You too, Soleil? Yeah, so, yeah, I'll just get into it then. Just a moment. My sister is the DJ. Science, the final frontier. These are the voyages of the Jones Lab. Our 5-year mission to explore strange new ideas and seek out new cures and new mechanisms to boldly go where no immunologist has gone before. So that's sort of what we do in the Jones Lab. We call ourselves the Awesome Jones Lab because we have a lot of fun doing our science. So HIV and AIDS has been around for a lot longer than we actually know. Its public endeavor has been since the 80s, since 1981. Since the beginning of that pandemic, 75 million people have become infected with HIV, and 32 million people have died of AIDS-related illnesses. And just as of last year, there are 38 million people living with HIV, 25 million are accessing the therapy that treats HIV. And while that's a worldwide feat, that's incredible, and it takes a lot of effort from government, from different funding bodies and the pharmaceutical companies that develop the drugs, but that still means that 13 million people wake up every single day potentially facing death from AIDS, and to me that is unacceptable. So there are also 1.7 million new infections and 770,000 deaths from AIDS-related illnesses last year, just to kind of put perspective to some of these other viruses or bacterial illnesses that you hear about going around the world. So can we cure HIV? I don't know if tonight I'll convince you that the answer is yes, but I don't need the microphone. I'm sorry. No, it's okay. But hello? Does this work? All right, so hopefully, if I don't convince you the answer is yes tonight, hopefully in my lifetime I can contribute to that cure with a few teams of our leading HIV cure scientists here watching tonight. So first, let me tell you a bit about HIV. HIV infects T-cells. There are two main types of T-cells, CD4s and CD8s. They're the soldiers of our immune system, so we need them to fight infection. The problem is HIV infects one of these types of CD4 T-cells called CD4 T-cells, so they're essentially infecting the soldiers of the immune system. And what happens is that over time, pretty quickly, before someone even knows they're infected or is being treated for the infection, these cells, many cells get infected, the viral loads go way up, and these red cells here are productively infected cells, and essentially those cells start to die because the virus kills them or the immune system kills them, and when enough of those cells die off, enough soldiers are gone, and then a new infection comes in, any bacteria, virus, fungus, or even cancer, and we call that an opportunistic infection, and that's when you have AIDS, because that infection will end up killing you. So HIV indirectly kills you by killing your immune system, and then another infection kills you because you have no soldiers left to fight. So one of the things we have is the CTL Enterprise. So CTL is a cytotoxic T lymphocyte, so the other arm of T cells, which are CD8 positive T cells, and they basically can recognize these productively infected cells, these red ones that have active infection. They have these little antennae with proteins that HIV is making, and essentially this cell will say, put up its antenna with a bit of HIV and say, hey, help me, I'm infected, come and kill me. So that's exactly what these CTLs do. They develop a memory response against HIV, and when they see this, they can kill these cells specifically. And they're very specific, and they will only kill HIV-infected cells. As you see, choo. The problem is, this can control part of the infection. However, these cells become exhausted. So when these cells are exhausted, they don't have all of their function or capacity to kill the infected cells anymore. So we need more help to clear infection. So the development of antiretroviral therapy happened around 1996, and it is incredible, and over the last 30 years has become more and more incredible. And essentially, within a few months, the viral load will go away, the productively infected cells will go away, and essentially someone living who has access to antiretroviral therapy will have no productive viral infection anymore. This is a huge worldwide campaign, if anyone's heard of it, called U equals U, where undetectable equals untransmittable. So if someone is living with HIV, has access to their therapy, and has undetectable viral load, they can no longer transmit the virus to an uninfected person. So hopefully these massive studies worldwide can reduce stigma and discrimination against people living with HIV, to know that they are no longer, you should not be afraid of them. We already know that HIV is the transmission rates, or transmissibility, but now we know that someone who's taking their medicine won't transmit the virus to an uninfected donor or individual, which is amazing. So what's left? We have these CTLs, they're still HIV specific, but they're exhausted. We have the antiretroviral therapy, which is fantastic, but now we have HIV hiding in these cells. So like every good army, there's always a reservist army, a backup army to be called to attention when needed to help by the immune system. So these reservist soldiers, or resting CD4 T cells, are infected with HIV, but HIV is hiding inside them. That means that they're not detectable by these CTLs, they cannot see them, so they cannot kill them. And the antiretroviral therapy has already minimized their replication, so it's done its job, and it also doesn't see the hiding infection. We call this the viral reservoir, or the latent reservoir. And this is essentially the part of the reservoir that we're trying to get rid of in our lab, which proves more difficult than it seems. So these cells last forever in the human. Their half-life is long enough that it would take at least 70 years for antiretroviral therapy to get rid of these cells. But new research has demonstrated how they proliferate, they clonally expand, they make clones of themselves. And this is done by two different mechanisms. First, they need to proliferate for the lifespan of the individual homeostatically to survive, to maintain their population, or they can do so in response to antigen from another bacteria, virus, or cancer. One really cool example from a friend of mine, Simonetti, published that these clones were replicating in response to a cervical or a squamous cell carcinoma, actually a tongue cancer. And what was really interesting is because these are the soldiers, this soldier was told to fight this cancer. So when it started to fight this cancer, these cells expanded upwards of 900 million cells. So imagine you start with one, and then from a cancer happening in the body, there are 900 million cells now. So the interesting part is that when the cancer was treated, these cells sort of waned, so they went away. And the cancer came back, these cells re-expanded with the cancer. And this person, unfortunately, died of their cancer, and when they cut the CD4 T cells from the lesions, they were all these clones of HIV-infected CD4s. So there's many different ways that these cells persist, survive, expand, proliferate in our body. So that's one of the hardest things we have to fight as scientists, is figuring out how they expand, why they expand, and what we can do to mitigate their expansion or to kill them off. So some strategies for an HIV cure that we do in our lab and around the world have to do with the infected cells or have to do with the CTL itself. So for the infected cells, we want to make these clones visible so the immune system can see them, or we want to send them off to hide forever so that they'll never make virus again. To enhance CTL function, we want to make a stronger immune system, make those cells less exhausted so they can be reinvigorated, come back to life, and fight off HIV-infected cells, and we want them to expand at the same time. So in order to make the clones visible, there have been a lot of different trials in vitro, in vivo, in culture dishes, in animal models, and actually in human clinical trials. This mechanism is called shock and kill. So we want to shock the virus out of hiding so the immune system can see these cells because they become visible, and then they can come kill them. Unfortunately, this has failed across the board. The actual drugs that they've used are small molecules called epigenetic modifiers, histone deacetylase inhibitors. They work in cancer, but for a different reason. They do turn on HIV replication, but they don't turn on the entire cycle. They don't turn on every piece that the virus needs to make itself visible to the immune system. And that has been why these drugs have failed in the clinical trials. So I just had a paper accepted 2 days ago that investigates these mechanisms and why these drugs have failed from multiple angles. So to get into the super nerdy part, I work on viral splicing a lot. I'm really into RNA. And essentially, I demonstrated that the drugs used, these histone deacetylase inhibitors, prevent splicing from happening during reactivation. So the virus turns on, you have the transcripts of the full-length genome turn on, but in failing to induce splicing, you essentially don't get that antigen presentation. So that antenna that comes up, because those spliced transcripts make proteins that need to shuttle the longer RNAs out of the nucleus, the ones that would make those really important antenna proteins. And because this isn't happening, they don't present and they don't get killed off. So we know that these drugs increase transcription, but they fail to induce antigen presentation or even virus release from the cells. And those are two huge qualifiers for being successful. On the other angle, if you remember the CTL enterprise, these drugs also inhibit function of the CD8 positive T cell for similar reasons why they inhibit splicing of HIV during reactivation. So look out for that, JV. I think it's online tomorrow. So another strategy some friends of mine are working on. Has anyone heard of CRISPR-Cas9 technology? Yes, fantastic. So CRISPR is a gene editing tool that essentially cuts in a very specific piece of DNA, and it's quite on target. And some colleagues are trying to cut out part of HIV, the one that flanks the genome, so it'll be on the 5' end and the 3' end of the genome. And essentially when they cut it out, it will induce this non-homologous repair, but because they're cutting out two pieces, they essentially can excise the entire genome from the cell. And this has been quite successful in their culture, in their in vitro studies. Now they want to bring it to clinic, and they've done their first monkey trial. So the really interesting thing is that, or I think the most difficult thing about using this technology, of course, is delivery, because you essentially need it to get to every infected cell in every tissue compartment all over the body. Hi, Soleil. My little niece, sorry. She's so cute. So essentially, this delivery needs to get into all the tissue reservoirs, all the compartments where these latently infected cells are, because all it takes is one cell with a replication competent virus. If you miss one cell when they go off their therapy to see if a cure happened, that cell can repopulate the entire body of infection all over again. So the other caveat, which is kind of works for us, is that 98% of the latent reservoir is defective viral genomes that could never make infectious virus particles again. And while that's fine, we're really trying to target the replication competent virus, the ones that can make these infectious particles. Another strategy for cure is also gene editing, but using a different tool called a zinc finger nuclease. So something interesting is that HIV uses two receptors to get into the CD4 T cell. It uses CD4, and then it has two co-receptors, but mainly it uses this receptor called CCR5. So HIV envelope needs to bind there to trigger its, or put its genetic material inside the cell. And people that don't have this receptor, they have a 32 base pair deletion in this receptor. HIV can't enter their cells. So even if they were to get infected with HIV, they wouldn't actually get infected with HIV, so they wouldn't be their cells would never get infected, and essentially the infection wouldn't take hold. So by cutting out this receptor, HIV cannot enter their cells, so these zinc finger nucleases are another type of gene editing tool, another pair of scissors, and this is cutting out part of the host. So I kind of like the technology that cuts out part of the virus, because then that doesn't target the host, although these are quite good technologies. And again, cutting out CCR5 has been shown to be quite successful in inhibiting HIV infection. So something our lab focuses a lot on is actually the immune system side side of everything. And CTLs, so the cytotoxic T lymphocytes, they're the CD8 positive T cells that can target, recognize, and kill the infected CD4 T cells. And we have all of these strategies to make them stronger, to make them proliferate, and so that they're stronger killers for the immune system. And these are my fashioned nanoparticles. So essentially what we can do with nanoparticles is we can load whatever we want onto them, and we can target them to a cell type. So we can load nanoparticles. One of our favorite compounds at the moment is something called an IL-15 superagonist. So IL-15 has great effects on inducing the proliferation of these CD8 T cells and enhancing their cytotoxic capacity. So it makes more of them, and it makes them better killers of HIV infected cells. So we've done a lot using nanoparticles loaded to these CD8 T cells, and actually Tom here in the audience is our mouse guy. So we have this really cool humanized mouse model where we have a mouse that's born without an immune system. And what we can do is engraft CD4 T cells either from an uninfected donor or from an infected donor. And these CD4 T cells will engraft all over the body of the mouse. Then what we do is infect with a lab strain HIV virus, and mouse cells can't be infected with HIV, but these human CD4s that now are engrafted all over the mouse can be infected with HIV. So it's kind of a little avatar of infection for us that we can study our cure strategies. And we make these CTL clones from people living with HIV, and we can load them with nanoparticles with this IL-15 superagonist, and then we just also infuse those into the mice, and we watch as the HIV goes away. It's quite extraordinary to be able to use this mouse model. We're very appreciative of these little animal avatars we can use in the lab. So, a cure for HIV. Well, there has been 2 men cured of HIV in the world. Timothy Ray Brown, who is also known as the Berlin patient, who's a friend of mine, and the London patient, who is yet still chosen to remain anonymous. So in 2007, Timothy Ray Brown was cured, and it took 12 years to replicate that in another person. So the cure is really interesting. First of all, both of the people had cancer, and I think the fact that they had cancer was actually part of their cure. So they both needed a stem cell bone marrow transplant, so they had to go through that really intense process. And in 2007, the doctor treating Timothy Ray Brown said, well, why don't we find someone who has that CCR5 deletion? So that receptor I said that gets cut out, so that the cells that repopulate his body can no longer be infected with HIV. So that's exactly what they did. Some 387 bone marrow matches later, and you imagine you're already lucky if you found one match. It took that many matches to find one who was also CCR5 double negative. So this mutation is very rare, and unfortunately for the world of cure, it's very white. So the HLA match, to have this match someone living with HIV, even they've done these studies in Spain, people living in Spain are too far removed from this Northern European genotype that you need to be able to match with these special. But the donors are incredible, so he had a bone marrow transplant. He got such severe graft versus host disease, he almost died. But then again, he kind of needed that. His graft needed to take over. His graft didn't take over, and any host cells remained, they could be infected with HIV again. So he had total body irradiation. He nearly died. His cancer came back. 2 years later, he went through the whole thing again, and he suffered a lot. He nearly died from the transplant. He nearly died from the radiation. But he's still alive, living without HIV. Incredible. And so they were able to replicate this in quite a similar manner just last year. So I know it's few and far between, 12 years later, but this gives us hope that we know it's possible. So we know we can do this. We can cure HIV. But of course, my passion is to cure not just 1% of people living with HIV, but everyone. So I hope that we can come up with something in my time in the lab, and as I transition to a PI one day, hopefully, maybe, that we can do this. So yeah, something we do here at Cornell, one of our cool clinical trials, we have this huge Martin Delaney collaboratory called BELIEVE, which stands for Bench to Bed Enhanced Lymphocyte Infusions to Engineer Viral Eradication. And essentially, this is a collaboration that spans labs in the United States, Canada, Mexico, Brazil. We have incredible collaborators. We get to meet once a year, and together we all work really, really, really nicely together, and that is definitely witnessed through my PubMed record of collaborating and getting to publish with them, working on studies with them. So one of the clinical trials we're doing in this, we already have 2 donors infused. So we take someone living with HIV, we do a blood draw, we remove the white blood cells from their blood, and we culture them with HIV peptides. So essentially everything that those antennas were presenting to the immune system to be recognized, we culture those with their cells. So you have the whole milieu of cells in there that you need to give the CD8 T cells memory. The dendritic cells will bring the peptide over. The CD8 will be like, hey, what's this? Let's be friends. But essentially, you already have HIV-specific CD8s in there. So the ones that see their peptide be like, oh, I've seen this before. I got this. And it will do that expansion and proliferate and then get stronger because there's also IL-15 superagonist in there. The CD8s who have never seen the HIV peptides before will be educated to them. So they'll essentially learn to have memory to them. And then eventually, we put this stronger immune system and reinfuse it back into the individual. So 2 of our participants have been reinfused with their peptide pulse cells, so we will wait and see what happens with them. So essentially, 2 men have been cured of HIV. A cure for HIV might be an individual endeavor, and a cure for HIV will absolutely be multidimensional. Thank you.
Thank you. Really, really amazing.